So I think that I have to introduce our next speaker, Dr. Pierre VANDERZWALMEN. Pierre, are you with us today?
Yes, I am present. Yes. Hello.
Hello, Pierre. I'm very happy that you are with us today. Pierre VANDERZWALMEN is a dear friend from long time ago. I know him since long time. He's also a friend of our association Egypt EFRI. Dr. Pierre is a member of Belgian Society of Reproductive Medicine, a well-known figure in our field, and he has been a permanent external partner of VANDERZWALMEN since 1992, and he is the biologist who achieved the world's first pregnancy using testicular sperm. Yes, he's the one. And since 2004, he has developed imposed a closed vitrification method for oocytes and embryo closed vitrification that we know today, and it makes it easy for the average biologist to apply and comply with the EU directives. He is a strong member in the EFRI and in the cryopreservation of human gametes, and he has a lot of publication. So welcome, Dr. VANDERZWALMEN. Your topic today will be osmotic stress associated with vitrification of oocytes, a very interesting topic. So the stage is yours, Dr. VANDERZWALMEN.
Thank you, Professor Amer, for this introduction. I will start to share my slides. Okay, you get them? You can see them? Yes?
Yes, excellent.
Excellent. Wait, I will try to close my share. Okay, perfect. Okay, thank you for the organization committee to invite me and to present you some data on the topic of oocyte vitrification. So, which stresses do the oocyte have to endure during a vitrification process? And I will try to give you some suggestions to reduce oocyte stress during a vitrification process. So if we look back, I would say more than 15 years ago, there was we could read plenty of publication on application of vitrification for oocyte donation for different reasons. And one of the main reasons was creation of an oocyte banking. That means they had to handle oocytes from young patients. And the literature showed that vitrification was superior to slow freezing. We need less oocytes to obtain a baby. And there was no difference with fresh oocytes for different parameters. So everyone at that time was happy, and vitrification, I would say, was considered as the best method for cryopreservation of oocytes. And vitrification is defined as a process by which the disordered liquid state of solute is brought to a standstill and as a solid without crystallization during cooling. And the strategy to reduce the likelihood of lethal ice crystal formation is to expose to the biological material to high concentration of permeable cryoprotectant and to achieve fast cooling and warming. And I would say fast cooling as well with open device and as well with closed device. But the situation was totally different as when we had to manage afterwards oocyte from advanced maternal age. And we observed a drop in the results. And this is a nice study for the team of Ubaldi and Laura Rienzi, who compare the clinical outcome between fresh cycle and warm cycle. And what we observe, first of all, with fresh cycle, and this is normal due to the aneuploidy rate, that the ongoing pregnancy decrease with the increasing of the age of the patient. But when we analyze the group of the vitrified oocyte, we observed a drop, but the drop between fresh and vitrified oocyte was dramatically decreased from 22, for example, to 7%. That means that this reflects the fragility of oocytes for advanced maternal age, which have to cope with non-physiological conditions. And we have also observed this in a study comparing fresh oocyte and to vitrified in an oocyte donation group, what we have observed, even though quite good survival rate, we have observed a drop, a significant drop in the rate of top quality blastocysts in the fresh vitrified oocyte as compared to the fresh group. Also, in our center in Belgium, we observed also a drop of 44% of top quality blastocysts in the vitrified oocyte group. So the question is, which stress is induced by the vitrification technique? And if we analyze the different steps, we have different steps in the vitrification process, the exposure to high concentration of cryoprotectant, and we go to 5 to 6 molar. And that means more than 5,000 to 6,000 milliosmol. Then we plunge in liquid nitrogen, the storage, the warming, and the dilution of the cryoprotectant. And each step finally induces kind of stress to these oocytes. So the oocyte have to tolerate and adapt to this non-physiological condition that may induce permanent and lethal damages. And today we will focalize on this osmotic stress that we induce during the exposure to cryoprotectant. And this is an oocyte, and the oocyte is specialized in specific orema with low permeability to cryoprotectant, low surface to volume, a complex structure. And in fact, we have the membrane of the oocyte, plenty if the membrane of the oocyte contain aquaporine, and aquaporine are very important. This aquaporine are specific protein channel that permit the osmotic flow through the membrane. And that means that it controls the transport of water and cryoprotectant out and inside the oocyte. That means that translocation of aquaporine to the cell membrane plays a crucial role to keep and maintain continuously the osmotic pressure balance in this oocyte. That means that these aquaporine that are included in the membrane is a kind of dynamic control. So, and we have also next to this orema, the plasma, the metaphase plate, we have the organelle inside the oocyte and also the cytoskeleton. So all these parts of this oocyte are very important. And you can see oocyte in a physiological condition and oocyte in hyperosmotic situation. That means that during the hyperosmotic reaction, during the exposure to hyperosmotic situation, the oocytes swell and also shrink. So which part is impaired in this oocyte? And there was a nice study from Hosseini who has shown that the cytoplasm is more affected by vitrification as compared to the metaphase plate. And his conclusion was that osmotic and thermal stresses induce cytoplasmic insufficiencies. And this is the main cause for poor competence of vitrified oocytes, which in turn highlight the crucial role of mammalian ooplasm to support resumption of meiosis, fertilization, and embryonic development. So these external stresses may compromise the intracellular spatial orientation, functions, and distribution of the organelle, and may disrupt the microtubular and microfilament networks. So, and we can see here what the different events that occur during the fertilization process. I will not go to detail, but you know this very, very well. So, are there some, I would say, suggestions to reduce this mechanical and chemical osmotic stress? First of all, some advised oocyte activation. I will not enter in details here. We can reduce this intracellular cryoprotectant concentration, and we are still working on this at the present. But also we can control the addition of the cryoprotectant in a more and smooth way. So this is a graph that shows you how cells react to hypertonic cryoprotectant solution, and we can observe that first of all we observe a shrinkage of the cell due to the outflow of water, and after that we observe a swelling of the cell according due to the entrance of cryoprotectant together with a small amount of water. So during these steps, chemical stress may be induced, but also mechanical stress due to this swelling and shrinkage process. And when we perform oocyte vitrification, you know very well there are different ways to add the cryoprotectant by merging the drops, by adding the cryoprotectant continuously. And here is a way how we add the cryoprotectant or method we add the cryoprotectant in 5 steps. But then our idea was why not to reduce finally this osmotic stress by increasing the number of steps, but during the same time. And what we have done, we compare finally, this is the volume change of a cell during exposure to different cryoprotectant solution, increasing the cryoprotectant solution, and you can observe the increasing the shrinkage process and the swelling process for each solutions. And we observe this during this cinematographic analysis, and we have compared this to a smooth addition of the cryoprotectant. That means we reduce the concentration for each steps, but what we observed a kind of smooth addition. We have done this first for germinal vesicle oocytes, and we compare the 5 steps to the 9 steps. And what we have observed, first of all, the survival rate, because you know that vesicle germinal are very sensitive cells. We increase the rate of survival, 61 to 87%, but also we increase also the rate of resumption of this germinal vesicle oocyte to the metaphase 2 oocyte from 64 to 73%. We have started to do this on metaphase 2 oocytes, and what we have observed, the same survival rate, 5 to 9 steps, but the rate of top quality blastocyst was 2-fold higher after adding the cryoprotectant in 9 steps. So multiplying exposure to 5 to 9 steps with a constant addition of cryoprotectant reduce osmotic shock, significantly increase the survival rate and increase the number of metaphase 2. And I think that in the future, now there is a trend to try to adjust the cryoprotectant to the oocyte by using microfluid platform. Then the last suggestion is to give time to time. That means that there are several studies in the animal that have shown that oocyte need an extra time to restore the damage to the cryopreservation procedure. And in fact, if we look, this was our first way of vitrification of oocyte and performing the warming. That means that after the warming, we perform, I would say, the ICSI between 30 minutes to 2 hours. But then, according to the literature, we think that it may be better to restore the cytoskeleton by giving more time to the oocyte to restore the cytoskeleton after the warming. And we increase the time after warming to 3 or 30 to 4 hours. And what we observed using this process, we observed also an increasing in the blastocyst rate and the top blastocyst rate by giving more time to the cytoskeleton, to the organelle, to resume their initial architecture. So my last slide to conclude, the permeability of the plasma membrane to water and cryoprotectant is important for the tolerance of cells to osmotic stress and is crucial for cell survival during cryopreservation. Non-control osmotic pressures induces lethal mechanical stress or chemical stress. This will affect dramatically the aquaporin translocation to the cytoplasmic membrane, impairing the outflow and inflow mechanism. That means I didn't show the slides concerning this topic, but there are quite a lot of articles showing that if you disturb the aquaporin translocation, that means that you impair the osmotic exchange. So smooth addition of cryoprotectants is one option to reduce the osmotic stress. Give time to time after warming to restore the full architecture of the cytoskeleton is also beneficial. Thank you very much for your attention. Thank you.
Thank you, Dr. VANDERZWALMEN, for this excellent work that you shared with us today.